Please switch off your Good evening, everybody. A warm welcome to IBD Masterclass.
Can I start, Rupa? I can't hear you.
A warm welcome to IBD Masterclass at IBD ENC. Today's topic is important, particularly for the postgraduates who are watching it, particularly for all IBD specialists to learn as to why IBD is increasing in this part of the world. IBD, back to the basics. We have leading stalwarts. We have Dr. Ashwin Ananthakrishnan. We have Professor B.S. Ramakrishna to give the keynote addresses. To introduce Professor Ashwin Ananthakrishnan, I have with me Dr. Jayanti Reddy. And it's a surprise. I'm sure, Ashwin, you like what Dr. Jayanti has to tell you. Over to you, Dr. Jayanti.
Good evening, everyone. You can hear me?
Yes,
Am I audible?
Yes.
Okay. Welcome to all. Good morning, good afternoon, good evening to all the delegates who joined this particular important session on basics on IBD. We have with us Dr. Ashwin Ananthakrishnan, who is a son of this soil, graduated from the Jawaharlal Institute of Postgraduate Medical Education and Research, JIPMER, in 2003. He is the son of his proud parents, Professor Ananthakrishnan, who is with us in this meeting today, who is a renowned surgeon from India, and Dr. Shanti, an equally renowned pediatrician from JIPMER. Dr. Ashwin is currently a senior consultant gastroenterologist at the Massachusetts General Hospital. His major areas of interest are in the field of epidemiology and outcome of patients in inflammatory bowel disease, focusing more on diet and environmental factors, which he's going to highlight in his presentation today. He has come out with several predictive models that can be used to inform patients about the disease, the course, and the likely response to specific therapy in inflammatory bowel disease. Dr. Ashwin has several publications to his credit and has authored several chapters in textbooks. Incidentally, Ashwin, I was there on the lighter side at your wedding reception at Chennai at the New Woodlands. And of course, we have a surprise for you. I requested Shanti and your dad, Dr. Ananthakrishnan, to join us in today's session because last time they missed you and they said you told them that you should not be listening to my talk and not intervene in the faculty. So I've invited them as faculty this time so that they're able to hear you. Over to you, Dr. Ashwin. You'll be speaking to us on environmental factors, the development and prediction of inflammatory bowel disease.
We have Professor Ananthakrishnan Senior. We would all love to see you.
We would like to see you and Shanti, Dr. Anand.
1 minute. Yeah.
We are indeed honored, sir, to have you with us today. It's a proud moment for us to have both Ashwin and Ananthakrishnan Senior and Ma'am Shanti Ma'am for being here with us. I'm sure we're all proud of what Ashwin is doing. He's made India proud on the big global IBD scenario. Thank you so much for being here today. Thank you so much.
I should thank Jayanti for sending us the invite. Otherwise, we wouldn't have known about it. Jayanti is my classmate in school. We were together.
Sir, I would like to tell you that I am a graduate of JIPMER. I graduated in the year 1981-82.
I was there.
Yes, sir, I know. I was in Ashish Basu's batch, Ashish Basu, Purnendu Roy's batch.
Right. Nice to meet you after all these years.
Thank you. Thank you for joining us. Thank you. Over to you, Dr. Ashwin.
Thank you, Rupa. Thank you, Dr. Jayanti. It is an absolute honor to present at this really, really important meeting. I hope you can see my slides.
Yes.
Yeah.
So what I wanted to address in this talk is to look at how the environment affects the development of IBD, and then finally end with a couple of slides looking at prevention, because I think that is the next phase of IBD research, that's the next phase of IBD care, in trying to understand how we can prevent the emergence of IBD in regions where it is rapidly increasing in incidence. And I really look forward to learning from everybody in this panel as to their opinion, what they're seeing regionally as factors that are potentially contributing to IBD. Right. The reason we're talking about environment in IBD is that multiple countries across the world are seeing similar rises in incidence. The timeline varies. For example, this is data from the Mayo Clinic cohort in Olmsted County, which showed that in the 60s, 70s, 80s, and maybe even early 90s, that there's been a tremendous increase in incidence. Before then, the number of new cases were about 2 or 3 cases per 100,000 in the 40s and 50s. And so they were tremendously uncommon there. But since then, they've moved up to 10, even 12 cases per 10,000, which is an increase in incidence of about 6 over the past 50 years. And this rapidly increasing incidence is not explained by changes in genetics. The changes in disease incidence that happen with genetics are those that take several generations to happen. And so when you see this changing incidence of disease that's happening over a span of a few decades, that highlights the role of environment. A second figure that very nicely illustrates the role of environment is this cartoon from a review that's now almost 10 years ago. And really the important thing in this is that the authors mapped out the regions of the world based on those that already had established high incidence. And those are North America, Canada, Western Europe, Scandinavia, Australia, New Zealand. We know that this was established in the 60s and 70s as being the regions with the highest incidence of IBD. And this cartoon highlights that. But what I want you to focus on in this slide is the group that is in yellow, which are India, China, Japan, Korea, in the Middle East. These are countries where the incidence was not very high, but which are seeing the most rapid changes in incidence, to the point that the rate of change in these countries now exceeds that in the West. And these are two regions of the world that are very different genetically. Right? But what is interesting is that they are experiencing environments that are increasingly similar. And so that again highlights that environmental factors are critically important in the development of IBD. Right? As we see this environmental homogenization, and so these are pictures from different regions of the world, you change the language, and if you change sort of the architecture that's intrinsic to a site, the overall concept of modernization and westernization and all the changes that happen can pretty much be applied to any region of the world. You'll see pictures like this from Asia, from the Middle East, from South America, from Africa, and obviously from North America and Europe as well. And so as there's this environmental homogenization across the globe, we're starting to see similar disease patterns, again, highlighting the role that environment plays in the development of these diseases. That's why we're all sitting here, is to ask ourselves the question, is IBD an environmental disease? There are multiple different environmental factors that have been described in association with IBD, and we'll spend some time going over them. And we'll end up by looking at the relevance of these factors, particularly in emerging nations. So if you talk about environment, how can the environment possibly affect IBD? This is a cartoon of IBD pathogenesis that we'll perhaps discuss more as part of the panel. But if you look at the environment, there are a number of different ways that the environment affects IBD. The direct, most plausible link for how the environment affects IBD is through the effect on the microbiome. We know a number of different environmental factors that we're going to talk about are influencing the microbiome and induce shifts based on their exposure. The second way in which the environment may possibly affect IBD is through affecting the intestinal barrier. Environmental factors may either alter the integrity of the mucus layer, which is important for preventing crosstalk between your bacteria and your immune system, or they can directly affect your intestinal permeability. The third way in which the environment can affect IBD is directly through its effect on the immune system with the innate and adaptive immune response and the production of pro-inflammatory cytokines. So every part of what is the architecture of IBD can be influenced by the environment. If you look at which environmental factors are relevant, we need to take a step back and look at the entire spectrum of timeline of influences. Right from the time of conception to in utero exposures in the mom, to exposures that happen in infancy, in childhood, in adolescence, and adulthood can all play a factor in the development of IBD. And let's sort of march through this timeline, understanding the impact of these factors as they happen at these particular life stages. First, what are the early childhood influences that can affect IBD? One of the earliest exposures for an infant is breastfeeding and weaning. And there are several mechanisms through which how this could potentially affect your IBD incidence. We know that various components of the breast milk facilitate oral antigen tolerance. They are critical in establishing your early life microflora, which then translates into your adult microflora. And there are specific components in the breast milk that are specifically interacting with certain bacteria, for example, Bifidobacterium and Lactobacillus, which then on the other end can modify your risk of Crohn's disease or ulcerative colitis. This was a meta-analysis that was published about 4 years ago that looked at multiple studies across the world in the West and in Asia, looking at the association with breastfeeding and IBD. And what it showed was that consistently across both diseases, children who were breastfed for at least 3 months in infancy were about two-thirds as likely to develop IBD as children who were never breastfed. And if the duration of breastfeeding was extended for up to 12 months, the hazard ratio for developing Crohn's disease or ulcerative colitis was only about 0.2. Again, this is not directly causal in saying that the decrease in risk is because of breastfeeding, but you can draw pretty consistent conclusions that at least children who are breastfed are much less likely to develop IBD. A second important factor that has been demonstrated in most Western studies is the role of antibiotics. This was a nice study that came from the UK general practice research cohort. And what this group did was to look at antibiotic exposure in infancy. And what they showed was that compared to those who were not exposed to antibiotics, which is the blue line, children who were exposed to either 1 or 2 or more than 2 courses of antibiotics were significantly more likely to develop IBD. And you see the lines getting steeper, suggesting a higher incidence. But what was also really fascinating from this study, which also has direct implications for prevention, is that this increase in risk was greatest in children who were exposed to antibiotics early on. So if children were exposed to antibiotics before the age of 1, they were 5 times as likely to develop IBD. You see that the line never reaches 0. So even exposure in adulthood to antibiotics is relevant for the development of IBD, but the strongest effect size is seen when you're exposed early. A third early life exposure that's potentially of relevance is the question of sort of hygiene hypothesis. So this came up in the context of asthma in the case of atopic dermatitis, and various immune-mediated diseases, where a more hygienic environment and less exposure to germs is associated with increased risk of allergic and immune-mediated diseases. And this was summarized in this meta-analysis looking at exposure to pets during either childhood or adulthood, also showing that there is some benefit, particularly for exposure to pets during childhood, against the development of Crohn's disease or ulcerative colitis in later life. The exposure to pets and animals in adulthood is not quite as clear. And you can see here from the graph that the effect may not be similar across all populations, and we'll discuss that at the end as well. But that's another potential early life factor that is important in establishing the risk of Crohn's disease or ulcerative colitis. There are a number of other early life factors that have been studied where the consistency of association is not as robust. Some studies have suggested that greater birth order and number of siblings, for example, larger family sizes, is inversely associated with IBD. The mode of childbirth, vaccination, and early life infection, their relevance in the development of IBD is less clear. Moving on to the next phase of life, right? And in terms of adolescent influences, what is the potential factors there that may influence IBD? And that brings us to the most consistently replicated environmental factor, which is smoking. And so this is a meta-analysis that's almost 20 years old now, but multiple studies before and since have shown this association, that for Crohn's disease, current smoking increases your risk of developing disease by about twofold. And the effect of smoking on disease natural history remains the same, in that current smokers tend to have a more aggressive disease course. If you stop smoking, the effect size is less. It does not reach 1 for at least 15 to 20 years after you stop smoking, and that hazard ratio is about 1.2. And what is fascinating here as one of the most fascinating environmental factors is that the effect is very different in ulcerative colitis. But if you're currently smoking, you actually seem to be protected against ulcerative colitis, in that it is very unusual to see someone develop severe ulcerative colitis if they're a heavy smoker. On the other hand, if you stop smoking, that seems to increase your risk well beyond what would have been even for a non-smoker. So your risk is about 2 times more likely that you'll develop ulcerative colitis if you stop smoking compared to if you never smoke. But if you continue to smoke, you're very unlikely to develop ulcerative colitis. And why we see this dichotomy in effect of Crohn's disease and ulcerative colitis, and what's the mechanism for this almost rebound that you see with smoking cessation in ulcerative colitis, I don't think we know yet, but this is a fascinating observation. Another adolescent factor that's been of relevance is appendectomy. This has been described in multiple Scandinavian and other Western cohorts, that if you have appendectomy before the age of 20 for appendicitis, that is associated with a lower risk of ulcerative colitis. On the other hand, if you have either appendectomy at an older age, or if you have it for a non-inflammatory indication, you don't seem to be protected as much. Again, why that is, we don't know. This association seems specific to ulcerative colitis. And now we start talking about diet as a factor, because in adulthood and in late adolescence, one of the most important factors that we see as influencing risk of IBD and probably contributing to the emergence of IBD globally is that of diet. This was a study that we published a few years ago looking at high school diet in adults. So this was diet when they had between the ages of 15 and 18, showing that children who had a high intake of fiber in high school were less likely to develop Crohn's disease. Similarly, for those who had a high vegetable intake and a high intake of fish, and we'll see some mechanisms why that may perhaps play a role. On the other hand, children who were eating high animal fat or heme iron containing foods, for example, red meat, seem to be more at risk for developing IBD. And this is early life diet adjusting for the adult diet. So it's not because they were eating this in high school and continued eating it through adulthood, but that particular period seems to exert its own independent influence. Right? And finally, in terms of adult exposures, we're talking about long-term diet and behavior and how they modify your risk of IBD. One of the first studies that we did was to look at dietary fiber. There were case control studies prior to this that had shown that perhaps people with Crohn's disease and ulcerative colitis were less likely to eat fiber. But one could not conclude that that was a protective association, because it could just mean that people were symptomatic and not tolerating fiber and stop eating fiber. And so what we did was used a large cohort of about 80,000 women who were followed for 20 or 30 years. And they were consistently asked about diet and whether they developed Crohn's disease and ulcerative colitis. And this, I think, is an important factor that is relevant for prevention, is that a high fiber diet was associated with about half the risk of Crohn's disease. But what was particularly interesting in this study was that not only was fiber protective, but it seemed to matter what you're getting the fiber from. If you're having dietary fiber that is from fruits and vegetables, you see a strong inverse association with Crohn's disease. You don't quite see the same strength of protective association for fiber from other sources, including cereals and whole grains. So this seems to be a fruit and vegetable soluble fiber specific association that modifies the risk of Crohn's disease. Dietary fat is another macronutrient that's been described in association with IBD, and this is demonstrated by multiple studies both in North America and in Europe, and actually multiple studies from Asia as well, where a diet that is rich in M3 fatty acids, that's directly relevant to the fish intake during high school that we were talking about, seems to be associated with a reduced risk of ulcerative colitis. On the other hand, diet that's high in M6 fatty acids, such as red meat, are associated with increased risk of ulcerative colitis. So this sort of extends what we observed with the high school dietary pattern directly to polyunsaturated fatty acid intake. Those were individual macro or micronutrients in the food. What about broad dietary patterns? And so this very nice study from one of my colleagues using Swedish mammography registry data showed that patients or women and men in this cohort who were having a diet that is very similar to a Mediterranean, adherence to a Mediterranean diet, which is typically rich in fruits, vegetables, olive oil, minimizes red and processed meat, has moderate amount of alcohol intake. So that together constitutes a Mediterranean style diet. And the adherence to a Mediterranean diet is ascertained using a score called the M-Med score. The higher your score, the more Mediterranean-like your diet score, the more different it is from a Mediterranean-style diet. And so what this study showed was that women and men in the highest modified Mediterranean diet score, so a very Mediterranean-style diet, were about half as likely to develop Crohn's disease in adulthood compared to those who are eating a diet that is very different from a Mediterranean-style diet. So that provides some guidance as to dietary patterns. We recognize that it's not just about the components of the food, but how the food is processed as well. Because moving from unprocessed food to ultra-processed food, you see a number of changes. You have added sugars, added salts, added preservatives and emulsifiers. At the same time, there's a loss of nutrient content. And so this study, done by one of the students working with me, looked at ultra-processed food in about 240 participants who were followed for over 5 million years, showing that there was a significant increase in Crohn's disease with intake of ultra-processed food in the highest quartile, for example, Q4, compared to Q1, which was the reference group. So about twice as likely to develop Crohn's disease if you were eating a large amount of ultra-processed food, specifically if you were eating a lot of ultra-processed breads, frozen or ready-to-eat shelf-stable foods, sauces, cheese, spreads, and gravy, and some dairy-based desserts and packaged sweets. Those were potentially associated with higher risk of Crohn's disease. It was also replicated in another study using data from the PURE consortium, which includes data from low and middle income country participants as well, suggesting that this is not an association seen just in Western populations. The number of different dietary factors that you see here that may either affect Crohn's disease, may affect ulcerative colitis, may increase the risk or decrease the risk. And all of these are potentially important as we think about preventive interventions. not just about diet. This very nice study looked at stress and depression, showing that if you were depressed or had anxiety, you were about twice as likely to develop Crohn's disease, and this decreased with use of antidepressants, suggesting that perhaps treating your environmental risk factors is potentially beneficial in reducing your incidence, and this was similarly an inverse association with physical activity that was observed as well. Right, so the final two slides I have is, as we think about application of preventive paradigms to emerging countries where IBD is increasing in incidence, it's important to remember that not all environmental factors are universal. For example, for breastfeeding, the effect in West is that there's an inverse association. For Asian studies, there's a much stronger effect. So sometimes we might luck out in that some environmental factors have the same association in all populations, and you can directly apply preventive interventions from one population to another. But in other exposures, such as smoking, while it's consistently described to increase risk in the West, there's no effect noted on many studies in Asia. And so it's not clear that a preventive intervention that relies on smoking cessation is what is of benefit here. And for yet other exposures, such as antibiotics, you almost see an opposite effect, where in the West, it clearly increases risk. In studies in Asia, there have actually been some studies that have shown it is associated with a reduced risk of Crohn's disease and ulcerative colitis. So in the same preventive intervention may then not be translatable across populations. If from a Western standpoint, I'm thinking about preventive interventions, I would suggest we encourage breastfeeding when able to, minimize antibiotics, avoid smoking, adopt a high-fiber Mediterranean-style diet, and minimize processed foods. So if I had to come up with a set of interventions, this is what I would recommend. But again, we see there's heterogeneity in which populations may benefit from these interventions. We can then talk about in the panel, perhaps, who's the target of such interventions, whether it should be first-degree relatives, whether it's those who are genetically predisposed, whether it's those serologically at risk. There are studies that show that antimicrobial antibodies can develop 5, 10, even 15 years before someone is diagnosed with Crohn's or ulcerative colitis. So should we be screening based on those serologic markers and then targeting our interventions for prevention to them? Or should we do it with those with dysbiosis? If we come up with some quantifiable microbial dysbiosis, we target them for such interventions. And obviously, when should that intervention be applied? Is it at infancy? Is it at birth? Is it at early childhood? If you wait till adulthood, is it too late to actually prevent IBD? And I'm curious to hear what everybody on the panel thinks. And finally, what's the level of evidence required for such interventions, right? Is it feasible to have randomized trials of preventive interventions for a condition that is not terribly common? Do you extrapolate from secondary prevention studies where you use it to treat IBD or perhaps prevent recurrence after surgery, for example? Or do you look for surrogate biomarkers? Do we look for evidence that adopting these interventions reverses your microbial serology or restores your dysbiosis or reduces your serum proteomic markers before you develop the disease? And all of these are potentially important questions for us to consider as a field as we then talk about applying the preventive interventions. And so with that, this is my summary slide, just highlighting what we talked. I'll leave it up there, but I thank you all for your attention and very much look forward to engaging with the panel.
Thank you, Dr. Ashwin, for that wonderful presentation and giving us an update on the various environmental factors and the ways to go forward in preventing some of these factors, which may actually be instrumental for inflammatory bowel disease. I think we'll take up the questions after the second talk. So we have our next speaker, Professor B.S. Ramakrishna, who is fondly called as BSR and as Ramki by his peers. And he's currently the director and a senior consultant in gastroenterology at the Institute of GI Sciences and Transplantation at SIMS Hospital, Chennai. He's an alumnus of Christian Medical College, Vellore, and he joined the faculty of CMC and superannuated as the head of the Department of GI Sciences. He's a great scientist and enjoys unraveling the basic sciences of various GI disorders like IBD, inflammatory bowel disease, that is, irritable bowel syndrome, chronic small bowel diarrhea, gut microbiota, to name a few. He is one among the Indian researchers in the 2% list, and he's credited with this. Dr. BSR has several publications in national and international journals. and his overall citation exceeds 9,000 with a H index of more than 55. With these few words, Dr. BSR, we request you to kindly make your presentation on gut microbiota and IBD, from dysbiosis to therapy. Over to you, Rama.
Thank you, Jayanti, and thank you to Rupa and the IBD ENC for asking me to talk about this subject, which is fairly close to my heart. So if I may just share my screen now. Not able to hear you. I'm having a little bit of trouble here, but hello.
Yeah, now it's visible, but the other screen is also there. For sure.
Yeah, I'm
I think there's something with the three
a
dots.
little bit of trouble here getting this. Okay. So the gut microbiome and IBD from dysbiosis to therapy is what I was asked to talk about. And so let me start with this. So basically, I'm going to start by talking about, assuming that, at least some people in the audience would have forgotten their basic biology, and I'm going to start with what the microbiome is. So if you look at the tree of life here, this is Carl Lewis. Basically, you have bacteria and archaea at the bottom, and then we have protista, lactate heading out this way, animalia where we are, fungi, and so on. And many, probably centuries ago is really when Carl Linnaeus created taxonomy. So just to remind people, so we have kingdoms, then phyla, then class, order, family, genus, and finally species. And in the case of man, we are phylum Chordata, genus Homo, species Sapiens. So similarly, bacteria, we look at these phyla, and eventually we look at genus and species. That's really what is important to most gastroenterologists, that we need to understand that. All right. So what happened about maybe 30 years ago was that soil biologists or soil microbiologists started investigating bacteria in the soil. And a lot of what we do today in GI microbiology originated from that. So bacterial taxonomy now is based on analyzing what is called 16S ribosomal RNA gene. And this gene has some areas which are highly conserved. So let us say it is conserved across the entire kingdom, let us say maybe the green. And there are areas which are conserved across phyla, let us say maybe the black. And then there are areas which are conserved only amongst a specific genus, let us say the blue one here. And then you have those which are very specific for certain species, let us say this orange one here. So essentially what we do is that we try to look at the 16S ribosomal RNA gene, we look at some variable regions, we sequence that, and then we assign taxa, or which is the equivalent of the species. We now call them taxa based on this. So when we look at fecal bacteria, this is just representative of what we'll normally have. You look at this kind of mauve side or mauve pinkish side here. These are basically Firmicutes. Here we have the proteobacteria, which are green here. The Bacteroidetes, which are the blue ones here. And actinobacteria, which are shown in the purple wedge here. So these are basically 4 major phyla of the gut bacteria, which we need to think about when we look at microbiome studies. So when we look at fecal bacteria by phylum, and we look at India versus the West, and I'm showing you our data here versus some representative Western data which came from original studies in the US. So these are the basic important phyla: Firmicutes, Bacteroidetes, Proteobacteria, Actinobacteria. In India, Firmicutes predominates. Also, they're very common in the West. It's actually a much smaller proportion here. Why is this important? So the Firmicutes contains almost all the carbohydrate fermenting bacteria, the important carbohydrate fermenting bacteria, which provide short chain fatty acids and so on. Bacteroidetes have some species which are good and some which are harmful. Essentially, the Firmicutes, many of them are thought to be beneficial. The Proteobacteria are, kind of semi-aerobic, or they are facultative anaerobes, and they include bacteria like E. coli and Enterococci. And the relevance to humans is that they include many pathogens, including Vibrio cholerae, Salmonella, and so on. The Actinobacteria, which are actually, if you look at it here, they're very much less common in Indians compared to Westerners. They contain the Bifidobacteria, which are often a component of probiotic preparations. So what is dysbiosis? If we think of a balance between protective bacterial species and aggressive bacterial species in health, there's a maintenance of a balance. When that balance is tilted, you end up with what is called dysbiosis. And why is dysbiosis important? So here is just a cartoon which is taken from O'Hara's ENBO reports, the Fergus Shanahan's work some time ago. Essentially, these, if you look at these as the intestinal epithelial cells, they have what is called these PRR, or pattern recognition receptors, on the microvilli here. And these bacteria, these orange things here on the surface, overlying the surface, they are bacteria. The pattern recognition receptors recognize motifs, very small molecular weight molecules from these bacteria. Dendritic cells, which are underlying the epithelial cells, they also put out processes and sample the bacteria. Eventually, these dendritic cells, the antigen processing cells, they condition the naive T cells to form either the effective T cells, which are the Th1, the Th2, the Th17, the Th9, or to make them as T regulatory cells, which effectively provide a balance between inflammation and no inflammation. This is another way of looking at it. So this is just borrowed this cartoon from somebody else, acknowledged at the bottom here. So basically here, these are the luminal factors: hygiene status, pollution, culture, ethnicity, cohabitation, blah, 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 diet. And then they all influence the gut microbiota. The gut microbiota have regulatory species, which we talked about as protective, and inflammatory species, which we talked about as aggressive. And over a predominance of the inflammatory species leads to dysbiosis. It tilts the immune response away from the healthy regulatory T cells and the regulatory B cells to a Th1, a Th17, a Th2, or a Th9 kind of inflammation. And these cytokines, they go accordingly. So what are some indicators of dysbiosis? How do we know that the person has a dysbiosis? One is if there is a reduced diversity of bacteria. Now, if you think of India as having, say, 40 states, let us say, individuals from each of these states is different from another state, and that's diversity. Now, if you homogenize everybody into one and say there's only one kind of Indian, that's reduced diversity. So that's what we talk about when you talk about reduced bacterial diversity. There may be a reduced number of probiotic bacteria. For example, bifidobacteria and lactobacilli may be reduced. There may be increased pathogenic bacteria, example, enteroadherent invasive E. coli, which are important in a specific form of ileal Crohn's disease. Or there may be something which is a reduced Firmicutes to Bacteroidetes ratio. I told you that the Firmicutes, a large proportion of it are fermenting bacteria. They're good for us. The Bacteroidetes, some are aggressive, some are beneficial. But when there's a reduction of this ratio, it leads to greater inflammation. So for instance, just to give you some examples, this is This is a gel electrophoresis of 16S ribosomal RNA from a number of different bacteria, which are from a different number of Clostridium leptum from a number of different IBD patients. So, and controls. So, if you see more numbers of bands, it means that there is greater diversity. There are a greater number of Clostridium leptum strains. So, this has more bands. This has more bands. The ones with the less bands tended to be among patients with IBD. So, there's reduced diversity. There are methods of calculating diversity, the Shannon index, the alpha index, and so on. And when we look at where they're distributed, we find that patients with Crohn's and ulcerative colitis have less bacterial diversity than controls. There are other ways. So this is called a Sarkos diagram. So you have Crohn's disease patients here, you have ulcerative colitis patients here, and this here would be the control. So when you compare the Crohn's disease with the control or the UC with the control, you find the ones which are in blue are down-regulated in Crohn's. The ones which are in pink are upregulated in Crohn's, or maybe it is vice versa. I'll have to look at the image. See, no, it seems to be what I said is probably correct. So there are differences that you can identify. So there's another way of identifying dysbiosis. So there is a feeling that, if you look at fecal microbiota, it may not necessarily be representative of what's happening at the mucosa. So we looked at mucosa-associated microbiota and IBD, and here's a list of, here's a sample of what happens with mucosal microbiota. There are changes in patients with Crohn's disease and ulcerative colitis. Importantly, what we see here is that the Firmicutes to bacteroidetes ratio is reduced in patients with Crohn's and ulcerative colitis. So we can catalog these microbes as being good or bad. So here's just a list of them, and then you have to understand that these are things which are a matter of flux sometimes, but generally Bifidobacterium, Lactobacillus, the Clostridial clusters, which the carbohydrate fermenters, the Rosburia also and Eubacterium, also carbohydrate fermenters. Calicobacterium prostige, also a major short-chain fatty acid producer. Bacteroides theta iotomicron, Akkermansia are important good bacteria. And then there are a huge number of bad bacteria. If we look at some gut microbiome changes in IBD in India, in the stool we show a reduction of Clostridium leptum in Crohn's, of Clostridium leptum and Clostridium coccoides in UC, an increase in Clostridium difficile in patients with UC, and this accounts for the overgrowth of C. difficile and the presence of C. difficile associated disease in some patients with ulcerative colitis. When we look at the mucosa, here are some of the changes in Crohn's. Some of those hold good for patients with ulcerative colitis. And there's an increase of some of the bad bacteria in Crohn's disease in the mucosa, and so on here. I won't dwell too much on them because they may differ between different studies. What about microbiome targeted interventions in IBD? So this gentleman actually won the Nobel Prize for discovering phagocytosis. He's called the father of probiotics. So he was very concerned with reversing dysbiosis. He thought people aged and died because of dysbiosis. So he said, replace the putrefactive microbes with a healthy flora. So some ways of doing this over the decades have been colon cleansing, diversion of the fecal stream, antibiotics, probiotics, and fecal transplant. Let me just go quickly through that. So colon cleansing, this is a place close to where I work in Vadapayani. So this is colon hydrotherapy, and it is done by experienced doctors at affordable rates using imported equipment. Fecal diversion is something that we all have to resort to occasionally in an individual with complex perianal fistulizing Crohn's disease. Now, what this paper says, we're looking at a totality of fecal diversion patients, is that yes, it does reduce inflammation, but unfortunately, you can reverse the fecal stream only in 20% of the patients. you've done a diversion. The other 80%, you have to continue to keep the fecal stream divert from the area that is diseased. Antibiotics. So let's look at ulcerative colitis. 2 RCTs showed no significant benefit. Pouchitis, 2 RCTs showed benefit. Cipro was better than metronidazole. Crohn's disease, there are 24 RCTs. Antibiotics improve remissions, reduce relapses. The antibiotics which are used are ciprofloxacin, metronidazole, and rifaximin. What about prebiotics? So prebiotics are carbohydrates which are not absorbed by the bowel, the small intestine. They reach the colon, and the bacteria there use them to produce short-chain fatty acids and so on. And amongst this conglomerate of studies, let me just focus on one, which was a fairly moderate-sized study, about 105 patients in 3 arms, using isabgol, plantago seeds. They said that plantago was equivalent to mesalazine in maintaining remission in a patient with ulcerative colitis. Twice that ulcerative colitis. Now, probiotics in ulcerative colitis. If you look at this diamond here, and if you look at I'm sorry, I think I've gone on to maintaining this inducing remission. If you look at it here, it is heavily weighted by a multi-center study done by Ajit Sood and Vinita Ahuja at Delhi. 25% of the weightage comes from that. So that shifts it slightly towards favoring it. It's still the odds ratio, so it's still the p-value is still a little bit what is required for statistical. Maintaining remission, probiotics seem to be better at maintaining remission. The diamond here is a little bit more on the side of favoring probiotics. Again, it crosses 1, and therefore it is not, beyond doubt here. Probiotics in Crohn's disease, induction and maintenance of remission. Clearly, no benefit. It straddles the midline. Probiotics in Crohn's disease, preventing post-surgical relapse, which is a major problem, again straddling the midline. No benefit. What about fecal microbiota transplant? There are 4 randomized controlled trials. The details of the trials are shown here. They used enemas, duodenal administration, colonoscopy, and enema was more favored. Duration varied from 6 to 12 weeks, and the number reaching, so the endpoint was remission, steroid-free remission, or Mayo less than 2. And you can see there is some difference. So here's the meta-analysis showing that there is benefit. The relative risk reduced to 0.8. So clearly there is some benefit from fecal microbiota transplant in patients with ulcerative colitis. Now I'm just going to end with a little bit of speculation. So what causes IBD? Basically, it is an overly aggressive cell-mediated immune response to luminal commensal bacteria in genetically susceptible individuals. So you need an abnormal immune system. You need an abnormal intestinal microbial flora. We know that IBD develops in mice with specific immune defects in T cells. I'm not going to go through this. In experimental animals, if you take those same mice with genetic defects in immunity and keep them germ-free, they do not get IBD. Now, if you take these genetically deficient mice and add individual single bacteria, let us say E. coli, to them, they produce different patterns of colitis. Example, proximal colitis versus distal colitis. And we know that probiotics in experimental animals ameliorate colonic inflammation. Multiple studies to show that. We know something about the effect on T cell lineages, which are implicated in microflora-driven colitis. So where I'm getting to with all of this is that if you look at this Venn diagram, IBD is not one disease. IBD involves defects in immunity. It may involve defects in the epithelium, particularly in ulcerative colitis. may involve defects in bacteria. So UC is at this end of the spectrum, Crohn's is at this end of the spectrum. And depending on what mixture you have, you may have IBD-A, IBD-B, IBD-C, IBD-D, IBD-X. So here on the left is the microbiome chip that I and my group developed some years ago. So, and here we can genotype the host, and this is the genotyping specific for some of the polymorphisms which are seen in IBD. So what to propose is really that it is probably important to understand an individual's microbiome, an individual's genotype, and then develop specific therapeutics based on the individual. So that's basically just like pharmacogenomics. And this, in fact, has also been proposed formally by Dr. Balfour Sattler, who is the champion of Crohn's disease and IBD pathogenesis. And essentially what they said is that you do a fecal profiling, look at dysbiosis, targeted pathway, protective pathway, inflammatory pathway, do a mucosal profile for cytokines and so on. And then based on that, based on the individual, based on the microbiome, you decide which particular form of treatment to use. So I will end by saying that microbiome targeted interventions are helpful in specific situations in a patient with IBD. Currently available treatments need to be used cautiously and monitored. Integrating molecular data may better optimize management in the long run. With that, let me stop here. Thank you.
Thank you, Dr. Ramakrishnan. That was very good. In fact, both the talks, Dr. Ashwin and Dr. Ramakrishnan, very thought-provoking, have thrown up a lot of ideas, and that probably opens the doors for our session on the discussion. And basically for the discussion, we have these two talks as the seeds. And joining us in the panel for the discussion are Dr. Ravikant, who's quite passionate, I think, about genetics. Roop Jyoti, of course, a gut microbiome man. Partha and Vishal, who are going to add that clinical edge to the discussion. And of course, we have the privilege of having Dr. Ashwin as well as Dr. Ramakrishnan joining us for the panel discussion. So I am going to start by sharing the slides, getting to the roots of it. Okay. Okay. Now I am ready. Right. I hope my slides are visible, and I'll make it full screen now. Okay. So we have a lot of points for discussion. I think some of the points that I really noted down, and I think I was mighty impressed with the final few slides of Dr. Ashwin, where he said that there are 5 areas where intervention can be done. And based on whatever evidence that we have, breastfeeding, antibiotics, smoking, and then about the high fiber diet, particularly from fruits and vegetables, and minimizing of processing foods. We'll come to all that discussion later, but let us get to the root of this IBD discussion. And my first question is to Dr. Roop Jyoti. Roop Jyoti, Dr. Ramakrishnan has mentioned in great detail about the gut microbiome. Now, as a person who's worked in this area, and I would just like you to put it in a nutshell. We've been talking about microbiome for a long time, especially with reference to IBD. Now, which are the areas or which are the specific circumstances where gut microbiome plays a significant role in the treatment of IBD. Basically, I would like you to refer to these bench to bedside issues. Dr. Rupjot.
Yeah, thanks for the question. First of all, what we need to understand, and Dr. Ramakrishna has brilliantly described, actually, he had made my job much easier to discuss about the pathophysiology and then go on to the therapeutics. So, one of the important thing where gut microbiome comes into IBD is in the pathogenesis. Earlier, it was believed the chicken or the egg, and it's still the story at many forums that microbes, dysbiosis came first or the disease came first. But if we look at lab research, like when IBD was induced in germ-free environment, the mouse did not have germs, they did not have severe disease or did not have the disease at all. So that clearly shows that the microbes have a role in the development or in the severity of the disease. So that's the first thing, but that is very difficult to replicate in the clinics in humans. But if we look retrospectively in the human studies, after manipulating the microbiome or dysbiotic bacteria, there is an improvement in the disease by different modalities. So that indirectly is a good evidence that the microbiome is possibly inducing the disease at some point. But again, I don't think the microbiome alone is the factor. Again, Dr. Ashwin and Dr. Ramakrishna had already said there are multifactorial things, genetics, host genetics, and then the environmental factors. So it's an interplay of all these factors that leads to the disease phenotype that we see. So that's on the pathophysiology part. And once we understand that, that has led the way for therapeutics of microbial manipulation. And but again, there are several gray areas in this because if we talk from a global perspective, the microbiome for the Indian people, the normal Indians are totally different. Dr. Ramakrishna has a study, we have our study. When we compare it to the global microbiome, the normal microbiota is starkly different from the West, maybe based on the diet, genetics, whatever. So in the Indian context, how much microbial manipulation would improve IBD needs to be studied. So what we are quoting in all these meetings are Western data. We really don't know how this would pan out in the Indian population. So I won't go into the details of how this manipulation will improve. Already Dr. Ramakrishnan showed lots of meta-analysis. But my take is, I think we need more data on Indian patients and more research on the Indian IBD and the microbiome interactions.
Dr. Ramakrishnan, I reiterate the fact that, for long we've been discussing about microbiome with reference to IBD, but somehow even now as a clinician, I would say that my clarity of thought as far and perspective as far as microbiome and IBD is concerned is still not very clear, probably because we still don't have a large amount of evidence. Now, if I could try to put you in a corner, could I ask you, what are the gaps that need to be filled before we can say that microbiome has got a very important role to play in IBD?
Thanks, Jian. The trouble is that IBD is a little bit like diabetes. It's not little bit, it's very much like diabetes. It's polygenic inheritance. There are multiple genes, each of which may confer a relative risk of 2 or 1.5 or 1.2 in terms of developing IBD. So here you have a huge number of genes which may make an individual more prone to inflammation. And then you have this whole host of microbes, some of which may be bad and some of which may be good, which tend to skew the way your inflammatory system, your immune system responds to any kind of stimulus. So that is where the problem is. What we require eventually will be large data sets where you have a large number of very well, in those same people, we will need a very well genotyped gut microbiome. Eventually, analysis of that might tell us that, interaction of a specific microbe with a specific gene or a specific group of genes provides a particular thing, and then we can work to get around that. I think that kind of thing is a little bit further down. Of course, I will not predict that it will be 100 years away, because the rate at which knowledge and science is advancing is totally unpredictable. Somebody may come up with something out of the box, and we may have answers. But until then, I think for us, the easy way, as for us clinicians, the easy way is actually to modulate inflammation.
Okay, so more on this later. Actually, we I think there were a few questions, because a lot of our audience are actually residents and young clinicians, and as a clinician myself, having spoken about environment, genetics, microbiome, my interest basically is that I would like to know what are the key points in the immunology of IBD which every clinician should know or a postgraduate should know. Partha, could you just help us out with that, with a few simple, elegant slides which are easy to understand?
Yes. So the effect of this immunopathogenesis or the immune system in inflammatory bowel disease, that should be considered in the light of genetic factor, dysbiosis and environmental factors. They should not be considered alone. All these four factors should be there to trigger the inflammatory response in IBD. And this immune cell activation is the effector arm for the inflammation to occur. And this dysregulated immune response, there are different components like innate immunity, which consists of dendritic cells and macrophages, which are the first line of defense against any microbes. Then adaptive immunity, which includes humoral immunity and effector T cells or regulatory T cells or natural killer cells. Soluble inflammatory mediators, that is different cytokines, we all know about them, like TNF alpha or in different interleukins. Other pathogenic mechanisms like epithelial cells, mesenchymal cells, which are non-immune cells, but still they are important because they cause angiogenesis, fibrogenesis, as well as lymphangiogenesis, which is important for causing different complications like fibrosis, fistula formation in inflammatory bowel disease. Also, enteric nervous system produce different neuropeptides, and there is a role for obesity and mesenteric fat as well in Crohn's disease. And, several novel pathogenic mechanisms are identified like inflammasome, which are cytosolic proteins which induce this apoptosis in this immune response pathways. Regulatory RNAs have different different microRNAs, they have different regulatory effects. Also, this DAMPs, which are disease associated microbial patterns. I'll just touch upon that. Can you show my next slide? So this is in the innate immunity first line, which is the first line of defense for the intestine, and among them the first one is disease associated microbial patterns. Whenever there is a exogenous insult to the epithelium, there is this different immune cells are activated, and they activate the different non-immune cells like fibroblast, endothelial cells, which in turn again leads to tissue damage and release of this disease associated microbial patterns, which is a sterile environment. Again, this is a vicious cycle which goes on, and this leads to non-resolving inflammation. And this DAMPs actually, if you can know this fecal calprotectin, this has two components of S100 proteins. These are actually DAMPs only, and they stimulate this pattern recognition receptors, which are this toll-like receptors and nod-like receptors, which ultimately activate this macrophages, innate lymphoid cells and dendritic cells, which are component of innate immunity. And this dendritic, among the innate immune cells, the dendritic cells are important because they have dual function. They, in a well balanced intestinal homeostatic environment, that will cause a tolerogenic dendritic cells, which are regulatory functions, whereas less diversified microbiota, allergens and pathogens will lead to immunogenic dendritic cells, which activate the effector T cells and cause intestinal inflammation. Whereas macrophages in normal intestinal mucosa, it has an anergic signature and acts as a bactericidal and phagocytic activity, whereas in IBD it can have a M1 or pro-inflammatory activity. Show my next slide. So one more click, I think. Yeah, yeah. So the different components of adaptive immune responses are there. So I want to highlight the fact that the Crohn's disease is a Th1 and Th17 disease, whereas ulcerative colitis is the atypical Th2 disease. So and ulcerative colitis, the effects are mediated through Th17, Th2 and Th9 affected T cells, whereas for Crohn's disease, it is mediated through Th1 and Th17 affected T cells. So this Th9 is important for this barrier function. It impairs the epithelial barrier function. As you can see, I have highlighted the different cytokines that are important in different ulcerative colitis, Crohn's, and some are common for ulcerative colitis and Crohn's disease. And this is important because, say for example, I have highlighted TNF and interleukin 17 in different color because just to give an examples, like our JAK inhibitors like tofacitinib does not influence these cytokines. So if you use this, that's why you can explain why different drugs may not be effective in treating IBD in all the patients. And in the lower panel, I have shown the redundancy of the immune system. If you block one pathway, another pathway may still be effective. So that's why there is thought that we can block multiple pathways using multiple biologics, but there are concerns regarding the side effects. Also, there is plasticity of T cells, that this Th2 or the regulatory T cells, there is lot of infidelity and they switch among themselves. So that can also lead to dysregulated immune response. My last slide. So this is the IBD immunopathogenesis in a nutshell. So environmental factors, genetic factors, and dysbiotic gut microbiome, and then immune system, which leads to activation of this innate immune cells and adaptive immunity, which consists of different effector T cells, also with different regulatory RNAs, inflammasomes, enteric neurons, and also different non-immune cells like adipocytes, ultimately leads to production of different cytokines and chemokines, which leads to this inflammatory pathways in IBD. On the right-sided panel, I wanted to show that in a normal, in the case of intestinal homeostasis, when it is maintained, this commensal bacteria stimulate the epithelial cytokines, which leads to tolerogenic dendritic cells and macrophages, which leads to activation of regulatory T cells, which in turn lead to secretion of interleukin 10, which inhibits immune cell activation and differentiation and proliferation. Whereas when there is pathogenic bacteria, it stimulates different pro-inflammatory cytokines, such as which in turn it activates Th1 and Th17 and Th2 cells, and also Th9, which depends upon the cytokine milieu, which has effect on impaired epithelial barrier function. Also important is different innate lymphoid cells like NK cells and intraepithelial lymphocytes, which leads to epithelial barrier maintenance, which is important for homeostasis, but it can also lead to epithelial proliferation and neutrophil recruitment, and ultimately lead to downstream activation of pathways leading to chronic inflammation and then different complications like fibrosis and fistula formation. Thank you. So this is in a nutshell about the pathogenesis for IBD.
You would agree with me that even the nutshell is very complicated. I thought this is probably the crux of the whole thing, where you have the three factors right on top, which seem to be, they may be linked or they may not be linked to each other, but then ultimately what happens is there's something like an immune mediated problem or in the gut, which may be linked to any one of these pathogenic mechanisms. But I think this is the simplest way probably to understand this whole thing, and these are all very many players. Now, Vishal, I would like to bring you into the discussion, basically, again, going back to some specific factors in the environment. I referred to 5 of them, which Dr. Ashwin had mentioned very clearly towards the end of his talk. So what, as a clinician, clinical gastroenterologist, so what specific factors in the environment you believe would have the greatest impact on the pathogenesis of IBD? And you could take it up as Crohn's disease versus ulcerative colitis or IBD as well. Over to you, Dr. Krishna.
Yeah, I think after the lecture, I don't think there is anything much to add. I mean, everything has been covered. But if I were to, highlight the salient differences amongst the environmental factors responsible for Crohn's disease and ulcerative colitis, I think largely one, of course, is smoking and current smoking being protective for ulcerative colitis, and how it has been highlighted that once somebody stops smoking, there's a rebound. But for Crohn's, the current smoking may be, positive, or may predispose to development of Crohn's disease. The other, of course, is appendectomy, which somehow seems to, protect from ulcerative colitis, but it is not very clear what effect it has on Crohn's disease. And if you look over a long period of time, probably there's no effect. But over a short period of time, there may be a relationship with Crohn's disease, and one can't be sure that that is positive or not. The other, of course, is about the diet, and largely that epidemiological data is kind of similar, except probably for the fact that fiber, seems to be protective for Crohn's disease. Now, it's not entirely clear whether this reduced intake is, protective, or it's a result of, some kind of disease and avoidance of fiber. But the consensus would be that, fiber intake is protective for Crohn's disease, and low fiber predisposes to, occurrence of Crohn's disease. There's just a couple of things which, need attention. One is, of course, we are in a TB endemic, and there is some suggestion that John's bacillus, that is paratuberculosis, has a relationship with Crohn's disease. There are other organisms like adherent invasive E. coli, which may have a relationship with occurrence of Crohn's disease. And since we are in India, I think last one thing which I would like to point out is the north-south difference. Why in north, we have a lot of ulcerative colitis, and why in south, the Crohn's disease is much more frequent. That is still not very clear, although people do talk about an epidemiological transition, that in places where IBD has been for some time, there may be a transition from ulcerative colitis to Crohn's disease. But it's not entirely clear whether that explains the differences in these geographic differences in occurrence of Crohn's disease and ulcerative colitis.
That is quite brilliant. Ashwin, as an extension of your presentation, a very interesting point that you made was fiber from fruits and veggies or vegetables seem to be protective, whereas fiber from cereals don't seem to be protective. Now, I wonder whether it's all because of the antioxidants or something like that. How do you explain, or what comes to your mind when you seek an explanation for this observation?
I mean, it was an interesting observation. There are some other studies that have linked grains to lower risk as well. So I don't want to quite make it seem as separate as was on my slide. But there are a number of different factors. There are many ligands that are found in fruits and vegetables that specifically interact with the immune response. For example, vegetables like cauliflower and broccoli, they have ligands that bind to the aryl hydrocarbon receptor, which we know to be anti-inflammatory. So there may be certain components, not directly with fiber, but other parts of fruits and vegetables that confer that protective effect, and it may not be entirely mediated through fiber. Soluble fiber has a different effect on the intestinal barrier than insoluble fiber. And the effect on the microbiome is also different for soluble compared to insoluble fiber from grains. So there are sort of few different reasons. It may not be entirely as separate, but there are a few different reasons why the association may be specific to Crohn's or ulcerative colitis.
Okay, thank you. Okay, we've talked about environment, we've talked about microbiome. I think we've not talked much about genetics. And all of us who see a lot of cases of IBD would vouch for the fact that we are seeing a lot of family clusters of IBD. Dr. Ravikant, how do we explain this observation? What proportion of patients of IBD could be probably linked to genetic factors? And it was also mentioned by Ramakrishnan that, genetics, environment, microbiome, all of them could be linked together, if I got BSR right. So are they interlinked or are they separate? Ravikant.
from the genetics perspective, the clustering in the family points towards a genetic susceptibility, definitely. Most of the genetic susceptibility comes from familial studies, twin-based studies, and gene mapping. But if you take only the genetic susceptibility into consideration, it might not explain the complete picture of the inflammatory bowel disease. Definitely, as Dr. Ashwin has explained, there are a lot of environmental factors that are there. Like Dr. Ramakrishna has explained, there are a lot of variants that have small effects which are very common in the general population, but all put together, probably the entire risk might be higher. So to put it in simple terms, it is definitely the interaction between genetics, environment, and the onset of the disease is probably in a genetically susceptible individual with an environmental trigger.
Dr. Ashwin, would I be right in saying that the IBD that has got a genetic background to it is the one that presents much earlier in life?
So I think we have to separate out into two different types. There's this phenomenon of very early onset IBD, which is seen in those below the age of 5, frequently with sort of severe perianal disease, very therapy refractory. That is very strongly linked to individual genetic variants, for example, an IL-10 receptor, and that bone marrow transplant is actually very effective for that specific genetic form of IBD. For the vast majority of IBD, studies have shown maybe a slight difference in genetic predisposition based on age. People who get it below the age of 18 are, and we're talking very, very small differences. If you take the overall effect size of the variance, let's say it's 1.3 if you're below the age of 18 and 1.15 if you're getting it above the age of 18. So yes, there's a separation, but it's not very strong outside of that very early onset disease.
Ravikant, more out of curiosity, have you been able to identify some genetic abnormalities that are linked to IBD?
Yes, there's something called as monogenic IBD, where a single gene and a variant in that, which are the rarer variants which are there. Yes, they have been mapped excellently, but by and large, the inflammatory bowel disease is polygenic with more common variants that are implicated.
Okay. Now, so you mean to say there are no specific genetic markers, no?
The compelling evidence is with respect to the NOD gene and the COD15 gene, which is predominantly seen in the Western population. But in the Japanese and predominantly the Indian and the Asian population, this is not implicated. We don't see these variants to be associated. So probably there's some other susceptibility which needs to be identified in this part of the world. There are few studies that have actually isolated, again, the more common variants that are there with small size effects. But IL-10 receptor gene probably is one of the few genes where a variant, the rarer variant, is implicated in early onset of IBD.
Dr. Ashwin, in clinical practice in the States, what would be the frequency of genetic screening or genetic mapping of patients with IBD?
Very, very uncommon. So only in very early onset pediatric therapy refractory disease. Otherwise, in the adult practice, I have never sent a sort of genetic sequencing on any of my patients, unless there's sort of unique multi-system phenotypes that can be explained by single variants. But broadly, not very useful.
Okay, Partha, I'll just give you it's linked to this particular problem that we are actually discussing about genetic background. Suppose we have more than one case in a family. Now, what would be your advice to the rest of the family? Now, suppose it's a mother and with 3 children, one child has IBD, mother has IBD. Now, what about the other 2? What is going to be your advice based on whatever we know about genetics and IBD?
Yes, familial IBD is the strongest risk factor for inflammatory bowel disease, and it has been seen if both the parents are having inflammatory bowel disease, the risk of having the children of having IBD is almost 33%. Almost 1/3 will have inflammatory bowel disease. Specifically, the study which was done in Crohn's disease, if both the parents are having IBD, and the chance of developing in the child is almost 1/3. And usually we also see that the sibling pairs, that different sibling pairs are affected with IBD, and in those cases usually both the cases will be, the diagnosis of IBD will be within 10 years of each other, and almost 2/3 will be within 10 years of each other. And I have seen personally 2 cases in one girl along with his brother, both, and they will have both concordance in location as well as phenotype. So I have seen 2 patients in which I have seen both ileocecal Crohn's disease in the brother and sister, and ileocecal Crohn's disease with stricture in 2 brothers. So they have both have the stricturing disease, as well as the location was also same, this ileocecal region. And also interestingly, one of the patient had developed a severe leukopenia after giving azathioprine. I checked for NUDT15 mutation in the first one which was diagnosed. It was homozygous mutation was positive, and before starting the next case was diagnosed after 2 years, when I did also before, because he was also afraid of starting azathioprine, we did NUDT15 mutation testing, and he was also heterozygous for that. So that suggests there is a strong genetic role. And studies have shown that if there is 2 family members are affected in a family, this concordance of location and phenotype is almost 50%. And if more than 2 family members are affected, then concordance in location is almost 80%. There is also another factor which is called genetic anticipation, like in case of other familial colonic cancer, that almost the younger patient or the sibling usually develop the disease or much younger, at much younger age. Almost they have seen almost 2/3 develop 16 years earlier than the onset of disease in their parents. Almost 2/3 have developed like that, and this is not due to assortment bias, due to earlier diagnosis in the siblings, because some of the parents developed the disease after the disease was diagnosed in the siblings. Also, in our experience, we have done almost 3,500 patients we studied and published the data in intestinal research. But we have found in that was published in 2019, we found that the incidence of familial IBD was almost 4% in our cohort. Sir,
your point is well taken. BSR, in your vast experience, given a situation like this, would you or would you not recommend maybe a screening scopy or follow up with stool tests or fecal calprotectin tests for the rest of the family?
Thanks. I think definitely not a screening scopy. I think it might be worth screening them in terms of height, weight, nutrition. Might be worthwhile finding out if they have symptoms. If they have symptoms, then I guess it is fine to go ahead and do a fecal calprotectin, because that's non-invasive and I don't see an issue there. The thing is, in addition to what Dr. Pata said, it is not uncommon. I mean, I've seen patients in families in which one patient will have Crohn's disease, the other will have ulcerative colitis, and that's not unknown. It's actually well described in the literature. So it's a little bit difficult to understand where all this comes from. My understanding is that there are some genes which have a strong effect. They predispose you to have inflammation. And then there are other genes which have a weaker effect. And if you happen to have several of these, and depending on what bacteria you have, you may have different morphological forms of inflammatory bowel.
Yeah, I think that's good. We move on and we go back to the gut microbiome. I have put this question. It's a little provocative, and I did raise this point earlier on during the discussion that gut microbiome, our discussion has gone on for too long. So we've created a lot of noise, but has that converted itself into a action is what I would like to ask Dr. Roopjyoti. Therapeutic modalities aimed at gut microbiome. we know about the fecal FMT, or now known as IMT, the other therapeutic modalities aimed at modifying the gut microbiome. Do you think that we have some areas where we have concrete mode of action? Dr. Rupjit.
Yeah, I think this is a very relevant question, and you are very correct. Like so many years we had been listening, talking, like manipulate the gut microbiota, and there will be some benefit. I think till 5-7 years ago, there was not much headway, even though some studies were there. But looking at the recent literature and the meta-analysis, in certain specific areas, there could be some benefit that may translate to clinical benefit. For example, if we talk about fecal microbial transplantation and ulcerative colitis, there are 4 meta-analyses Dr. Ramakrishnan had alluded to, and 3 of them had some shown benefits. But the catch here is those benefits were specific to people. In one of the studies, which was a very good study, I would say, there is a concept called super donor. So only from one particular donor, the patient who got, they had benefit. And few other studies have shown that patients with a specific microbial signature would benefit more. For example, who has more proteobacteria or fusobacterium, they won't respond well. But patients who have more of Clostridium, there are different clusters. For example, cluster 4, they would respond better. So in a research setting, these are fine. We do it in a controlled environment. We have the fundings to do all these things. But when we want to translate it to clinical reality, we don't know what is the fundamental or baseline microbial signature of those patients. So what we see the percentage in the clinical, in the research setting, I'm not sure if we can get a similar percentage in the real life clinical scenario. For that, maybe we might have to do some preliminary microbial testing in this patient before starting, which again is very cost prohibitive. So how much this research data is translated to the real life clinical setting, I'm still a little concerned.
So a lot of gray zones there.
Yes.
Dr. Vishal, I know I did read your message about the fact that you don't have much of an experience as far as intestinal microbiota transplantation. But I just like to ask you, are we in a position to do 2 things? One is diagnosis of gut dysbiosis. BSR mentioned about diagnosis of gut dysbiosis. As a clinician, I believe that it is still very much in the experimental phase. I don't know whether it is relevant or logistic to think of a diagnosis of gut dysbiosis. What is your opinion?
Yeah, I mean, I have heard of commercial agencies offering some gut microbiota testing, but I don't know what to do with that, So at this stage, I think we are not there to really, use that particular piece of information. And as you said, although we are kind of now getting interested into FMT for IBD, but as of now, we have not been using this as a therapeutic modality for our patients.
Dr. Ashwin, have we moved over from FMT to IMT now? And has the day come when we can think of having these capsules as over-the-shelf purchases for this purpose? How is it
not yet? Case? I mean, I think this question, I'm glad to hear it comes up so many times in India as well. This question comes up a lot in my clinic, where the concept of sort of microbiome therapies is very attractive to patients. I think we have to separate out the relevance of the microbiome from our ability to change the microbiome, right? And that's where we're not there. I think just because we can't do the second, it doesn't make the first part not relevant. There have been very nice studies that if you take probiotics, nothing changes. Your microbiome doesn't change, your metabolome in your stool doesn't change, nothing changes. And your fecal transplant is the other extreme where everything changes. A lot of good things change and a lot of bad things change. In animal models, if you exactly transfer 1 or 2 strains, you're actually able to treat inflammation very well. We just have to be able to translate that in IBD. And there are actually some good trials that are starting in Europe, for example, looking at Faecalibacterium prausnitzii, which is a butyrate producer, very commonly depleted in IBD. In animal models, if you give it orally, it actually helps treat the colitis. If those kind of studies are translated into human interventions where you're meaningfully able to change your microbiome function, then I think current probiotics, just random pickup strains, random doses, not going to work.
Okay, that's great. So I will just skip this in the interest of time, because we know the answer still not very clear, and this is what was highlighted by BSR. And of course, a word about short chain fatty acids. I mean, obviously, it's linked to a lot of basic sciences in IBD. Do you think, Vishal, do you think SCFAs are worth a trial in patients with IBD?
I mean, I really don't know whether they are worth a trial. I mean, there were studies in 90s, randomized trials, some of them, showing benefit of butyrate in EMAs as an add-on therapy also in distal colitis. But really, it hasn't really caught up. I don't know whether it's with the preparation availability. But certainly, we do know that there is a problem with the short-chain fatty acid production. The levels are lower in ulcerative colitis. I mean, it is evocative. I don't know if somebody's used them clinically or not. I have not. But certainly, there is some evidence to suggest that they may have a role in, as a local therapy.
Your opinion, Dr. Ashwin?
I think we haven't been able to deliver it consistently to the effect. I think mechanistically it will be of benefit, and it could be a secondary product from either dietary interventions, for example, fiber, along with the setting of bacteria, generate short-chain fatty acids. So maybe we don't have to give short-chain fatty acids, but we give the raw materials that your microbiome can use to generate more short-chain fatty acids, which are then beneficial. So it's possible it's related to that.
Okay. So while I give each of the panelists a moment to think about one major takeaway which you would have from this session, Rupa, any questions from the chat box that you would like to raise? Dr. Rupa? Or Dr. Jayanti?
There is one question which has come in, and I'd like to ask Dr. Ramakrishna as well as Ashwin. The question is what is the difference in IBD between the East and the West? I mean, in terms of the disease or perspectives, maybe because Dr. Ramakrishna, you did discuss a little bit of microbiome difference between the East and West. So your perspective, and followed by Ashwin's.
Thanks, Rama. So from a long time ago, what we felt was that IBD in India was much less severe than IBD in the West. And I suspect, that this is something that as trainees and subsequently we felt that our IBD was much more easily controllable. And I think that kind of feeling has persisted. I don't think in terms of distribution of the disease or in terms of the other things, it is different. But as we've progressed, I think now we're seeing more severe forms of Crohn's disease, more severe forms of ulcerative colitis. And to me, that seems to be the major difference between India and the West.
Ashwin, your take on that?
I mean, I'm seeing only one side of the coin, so it's tough to say. But from the literature and from my experience in my practice, they seem similarly severe. So I don't think we can conclude that Indian IBD is milder. There seems to be slightly more common perianal disease among Indians, at least in the US, and from the literature, and whether that's biologically mediated or another reason, we don't know. Fortunately, the response to treatment seems similar. So even though the genetics seem different, our current treatments, in our experience, seem to work just the same.
I think, Ashwin,
which is reassuring.
That you say this, there is a very important point that I'd like to discuss. What we are seeing is, we are seeing a lot of migrants who are going to the US, and then, all these software guys who are developing IBD, they tend to have more severe disease than what we are seeing in India as such. So at the IBD ENC, actually we analyzed around 10,000 people data, and we found perianal disease actually to be low. Amongst extraintestinal manifestations, we found that, arthralgias were much more common than the others. Primary sclerosing cholangitis, other kind of complications appeared to be low. So there is perhaps that environmental factor, which is the difference. And, patients, the migrant patients, they tend to have a little more severe disease, and that's what we realized when we were analyzing our data. So I think the area that we discussed today, maybe environment and related thing, there is some sort of link, because across this region, biologic usage is much lower than that of the Western world. Despite that, we really are not seeing that severe disease, if you say. The colectomy rates are low, and Crohn's disease is showing an increasing trend in southern India compared to the north. So I think environment is playing a role, and that's the reason why you are seeing Indians in the US who are having more of perianal disease, perhaps. There's another question, and I think this is for Dr. Ramakrishna from Dr. Sudeep Chatterjee. He wants to know, in simple words, how microbiome plays a pivotal role in IBD, and do you think that that can be modulated? So the same question also has come from Dr. Rajendra Patel, that can probiotics help in IBD? So there are two questions based on that. And I think regarding the probiotics, we'll take everybody's take. Dr. Ramesh, what do you think?
Yeah, actually, I was thinking about this question, but then I thought I should not probe it further, because I don't think we have the answer to it. The use of probiotics is different for each person. I think we'll have 6 different opinions if we ask 6 people. When do you use probiotics in IBD? Yes, sir. Your take.
Thank you. So, in terms of microbiome, I don't think we're still quite there when it comes to saying that we can prevent it this way or that. But do keep in mind that what feeds the microbiome is what you eat, and depending on what you eat, and all the constituents of what you eat do induce changes in the microbiome. That's one of the major reasons for the environment contributing to IBD pathogenesis. So I don't think that at this point of time we have any answers as to what you can do to prevent IBD. What I do tell my patients probably is that the tradition, sticking to a traditional form of diet certainly seems to help. Even in India, we find a difference between urban India and rural India. People born in rural versus urban. So early life conditioning, diet, everything certainly plays a role. So, but with regard to specific microbes, I don't think we have the answers yet. In terms of probiotics, Ramesh is absolutely right. There's no single answer that fits all. There's no single probiotic that fits all. Some patients may be benefited by some probiotics.
This is another interesting question, Dr. Ramesh. It has just come, and this is from a very senior and well-known name across Asia, Professor Raja Offender Raja Ali. He just wants to know, and I think since Ashwin is here, that it would be nice if we can have a study which can show a specific gut microbiome signature from different parts of the world, or does it really, is it already there? So is there a signature gut microbiome according to population or region, and what is your take on that?
Is there a signature for IBD or a signature in general? The microbiome is very different in Asia from the West. It's very strongly influenced by diet. It's influenced by early life factors. There are very nice studies that have looked at people who are traveling from Asia to the West. They've sampled them before they left very soon after they reach the West, and then a few months after, showing that there's a change even within days of your environment changing. So I think it's tough to have one static microbial signature that will likely be true across different populations. I think it's different for each place. There are probably common threads across all, at least functionally. Whether sort of the bacterial family is different or not, it remains to be seen. But functionally, there are going to be similar signatures across the population. But it's going to be a dynamic thing. It's not going to be a static signature.
Okay. So I think we had run out of time. Finally, I just like to go across all the panelists. One point that you would like the residents, the clinical gastroenterologist who's attending this webinar to take home. I'll start with you, Ravikant. One point from your area that you would like your audience to take home.
Although all three are important, the genetics, the dysbiosis, as well as the environmental factors, I think genetic testing in families where more than one member is affected would be helpful. And very early onset IBD, probably genetic testing would be helpful.
Very good. Vishal, your takeaway message for the audience.
Yeah, I think one thing which I'll take is that environment, everybody would agree that it has some role. I mean, we can't change genetics, but we have the opportunity maybe to halt this epidemic by making, the rising numbers we can stop by making some of the changes. Maybe dietary, maybe interventions in the families, like which were pointed out in the last couple of slides by Dr. Ashwin, tackling smoking and factors which we probably can. And I think there may be a window of opportunity for us to probably reduce numbers to some extent, not to a large extent anymore.
Thank you very much, Vishal. Partha.
Sir, all the four factors in conjunction is important to trigger IBD, and also this familial history is one of the strongest risk factors for development of IBD.
Roop Jyoti.
Yeah, so firstly, one should not jump into using probiotic just with a diagnosis of IBD as a co-adjunct treatment. Secondly, I think which is more important, research now, if you see, all studies are done in isolation. Microbiome is a different study, genetics is a different study. I think there should be one big cohort where we have to see the thing in a holistic manner. That's how we'll understand the crosstalk between the different factors. I think that would be an important.
Rupa, you wanted to make a point.
No, I just wanted to tell all postgraduate and youngsters who are listening in today, you can see this is such an interesting area to work in. We need youngsters to focus on basic sciences, basic science research in gastroenterology, because that is where we will find answers, prevention, and possible cure for chronic disease like inflammatory bowel disease.
Yeah, doing something like what on the lines of what BSR and Dr. Ashwin are doing. BSR, your message. Don't talk about the microbiome, something else.
In fact, I will not talk about the microbiome. What we've talked about till now is basically to do with pathogenesis. It helps us understand why the disease occurs. It provides us opportunities to identify targets for future action. I do not personally use probiotics in 95% of my patients with IBD, and I see a lot of IBD patients. I still use immune modulating drugs because that is what will affect, and that's my message.
I think yes, I think this is what we tried. And Ashwin, I come back to you, and I would refer you to the final slide that you mentioned. 3 questions, and I would like you to give your answers. Who should be the target for intervention? You had given 4 choices. First degree relative, genetically predisposed, serologically at risk, or those with dysbiosis. What would be your answer for us?
My answer would be first degree relatives, and among the subset, those with seropositivity to antimicrobial antigens. That's going to be the highest yield population for intervention.
Wonderful. And when would you, in this group, when would you think that intervention is most
I
effective?
think
Obviously,
it should start in infancy.
early,
I think it's
infants.
very important that we have this discussion with patients, things like breastfeeding, early life antibiotic exposure, the parent has to start pretty much at birth. And so this is something I tell all my patients.
And the third question is, what is the level of evidence for such interventions? Do you think it's strong enough?
I don't think it's strong enough, but none of these have been shown to change the risk of IBD. They're sort of associated with it. So, but it's the best we have. It's not strong enough. They're good for a lot of different reasons. None of what I'm saying is, going to put you at harm for anything else. So there's absolutely no reason to not do it, right? And while we're waiting for better evidence to come.
I think well said, well said. Vishal wanted to make a point because he's sporting a huge smile on his face.
Because it's not costly at all. These are all easy things to do.
Very good, very good. I think we had a wonderful panel discussion. In fact, I would like to thank each of the panelists and of course the lead speakers, Ashwin and BSR, for a great session. In fact, as a clinician, I've learned a lot. I hope members of the audience who are there have also learned a lot as far as basics of IBD is concerned. I'd like to thank each one of them. Ravikant, Vishal, Partha, Rupjyoti, and of course, our lead speakers, Ashwin and BSR. And with this, it's over to you, Dr. Jayanti.
Yes, so I will actually say that every IBD ENC meeting, every month, we highlight one of our pillar countries and we try and highlight what is being done. This month, the theme is Bahrain. And I can't, Mahiba, are you there? So I have the honor of having my dear friend and representing Bahrain, Professor Mahiba Abdullah. Can we have the video, please?
My name is Dr. Mahiba Abdullah. I am consultant gastroenterologist from Kingdom of Bahrain. I would like to thank Dr. Rupa and the committee for giving me this opportunity to give an overview about IBD in my country, Bahrain. IBD was considered as a rare disease or non-existent disease in my country, but this is no longer is the case because our data showed that the prevalence of IBD is increasing and it showed that it is 26 per 100,000 cases with UC prevalence of 17 per 100,000 cases. And this report was reported in 2014. There is an increase of at least 4 times as many cases reported in the later years compared to the earlier years. The current cohort IBD in Bahrain appears to be following the general pattern of disease development as in the countries undergoing westernization. And with this is which is described as initial low IBD incidence followed by an increase in UC while the CD remains low. This is then followed by an increase in CD cases, which is slowly approaching the UC incidence. And this pattern is similar to other countries like Japan, South Korea, Singapore, and Saudi Arabia. We have noticed that ulcerative colitis is more common than Crohn's disease with male predominance, and the hospital admissions were less in the earlier years compared to the later years, and with more severe and aggressive phenotype of IBD patients with complications like malabsorption, anemia, intestinal obstruction, stricture, abscess, and fistula formation, requiring surgeries on their first presentation to us. So we can conclude that IBD is no longer considered a rare disease in Bahrain. The incidence of both UC and CD is steadily increasing over time. The dramatic increase in the prevalence and incidence of IBD in Bahrain highlights these disorders as one of the major emerging disorders in the newly westernized countries. Efforts should be made to inform and educate public about the signs and symptoms of the disease, and more studies is required to better understand the pathogenesis of this disease. Thank you.
Thank you. Thank you, Mahiba. I think it's the same story across the region. And once again to say, we are now 22 countries and more than 1,000 members strong. I think we will have some answers about why the disease is increasing from this part of the world where it is all emerging. And over to you, Dr. Jayanti, for the closing remarks. And
thank you, Rupa. On behalf of Dr. Rupa, our Post Director for the IBDENC, and my co-chair, Dr. J. N. Ramesh, we thank our guest speakers, Dr. Ashwin, Ananthakrishnan, Dr. B.S. Ramakrishna, and all our panelists, Dr. Jyoti, Dr. Partha, Dr. Vishal, and Dr. Ravikant, for the wonderful session and taking us through this rather difficult topic. And you've made us understand some of the basics of IBD as well as some of the preventive measures that are involved in management of patients with inflammatory bowel disease. Over to you, Rupa, for the next session, details of the next session.
Thank you, Dr. Jayanti. And the announcement for the next month's masterclass, IBD Radiology, What's New? IBD Radiology, What's New? The date is 24th June, same time, Friday, 6:30 p.m. India. I hope all of you will join in and learn all the latest. No academic meeting is complete without thanking our academic partners. We have Janssen, with Dr. Reddy Labs, Micro Labs, and Takeda. Thank you. Thank you for your support. We have completed more than a year of IBD Masterclass, and I think with this education and awareness of IBD, we can increase across the region. Do send in your comments. Log into www.ibdenc.org and learn more about this chronic lifelong disease. Thank you, everybody, and good night.